or cell type. Now, this idea of preserving cell identity has also come about in the study of induced pluripotent stem cells. So P53 appears to be an incredibly strong inhibitor of the conversion of, in this case, a fibroblast into an induced pluripotent stem cell using the Yamanaka factors. Now, this is an incredibly important point given a number of potential therapeutic applications we might want to use induced pluripotent stem cells for, and recent observations suggesting that, for example, induced pluripotent stem cells often have P53 mutations in them, and they seem to be the mutations we find in cancer. So when we try to select for cells that have properly reprogrammed, oftentimes they will have mutations in P53. Now, unsurprisingly to me, although not to everybody, is that a recent paper came out suggesting that Cas9 expressing cells, or those we may want to use for genome editing purposes, oftentimes will select for cells that have inhibited or inactivated P53, such that when we want to make a particular genome edit, oftentimes we'll end up with cells that have our edit of interest, but also will have mutations in P53. So these are questions and different phenotypes that we really have to understand better if we want to use these therapeutic approaches in the future. Now, I'm going to apologize here for a moment because P53 is sadly a DNA binding transcription factor. All right, so P53 primarily functions, and most of what it does is done because it can bind to DNA as a homotetramer. Now, P53 can bind to promoter elements to activate transcription of numerous genes, both protein coding genes, and as we heard before, long non-coding RNAs. But we're really interested in P53 binding to enhancers, where again, it binds as a homotetramer, interacts with promoters, and can also stimulate expression of these small enhancer RNAs. So our real interest in P53 came from some fairly old observations, almost 30 years old, where P53 can control alternative cell fates to the same exact stimulus and at the same dose. So this is my terrible example of a fibroblast cell or a mesenchymal cell type. If we treat that cell with certain doses of IR, it will reliably and reproducibly undergo permanent cell cycle arrest or senescence. We can take, for example, a skin keratinocyte, or what we'll study a lot in our lab, our mammary epithelial cells, introduce the same dose of ionizing radiation, and this cell will almost always undergo the process of apoptosis or cell death. So we really wanted to understand, and these two processes in this case are completely P53 dependent. So we wanted to understand what mechanisms control these differential P53 dependent cell fates. So we undertook an RNA-seq and genomics approach to understand potentially how these different cell fates might come about. And we used both steady state measurements of RNA abundance, but also used a number of different approaches, such as the localization and activity of RNA polymerase II, the local chromatin structure, and also things like the expression of these small enhancer RNAs to really focus in on what are direct P53 targets, as opposed to mRNAs that perhaps undergo post-transcriptional regulation or changes in RNA stability. So what we noticed, really, in the summary of this, is the P53 dependent transcriptome is highly cell type dependent. Now, this was a bit of a surprise to us, to be honest with you. So we saw that there were genes, certainly genes that were shared. About 100 to 150 genes were commonly induced or P53 dependent. But we noticed that there were about 50 genes that were P53 dependent only in dermal fibroblasts. But we saw a massive expansion of genes, P53 dependent genes in mammary epithelial cells. Now, this is also true in other cell types we tested, such as dermal keratinocytes, which are another epithelial cell type. Now, when we looked at what genes these were and why there was this expansion, we noticed that the genes that were common to both cell types seemed to fit into that canonical model of P53, the idea that P53 can inactivate apoptosis, senescence, and some other processes. Now, it was interesting when we looked at the mammary epithelial cells, we saw a new set of P53 target genes, one group of which was actually epidermal development. So P53 in this cell type was able to activate a number of epidermal genes, or genes involved in epidermal development and lineage commitment. Now, we want to know the mechanisms that would let this happen. How can we get this massive expansion? Remember, p53 is expressed in both cell types. So how does it activate different sets of target genes? Well, the answer turned out to be fairly straightforward, which is that p53 bound or engaged with the genome at a number of new locations in mammary epithelial cells, here in a heat map, compared to our dermal fibroblasts. Now in these heat maps, each row is an individual p53 binding location. There were something like 5,000 unique genomic engagements in these cells. We can then compare where those locations are in the genome, and it turned out that they reflected what we came to realize were cell type dependent or cell type specific transcriptional enhancers, as evidenced by the patterns of histone modifications, which are reflective of that type of genomic regulatory element. I think it's clear to see that we have unique binding of p53 and a unique genomic environment at those binding sites. So this is a good opportunity to take a step back and remind everybody that transcription factors control and are controlled by chromatin accessibility. So this is a pretty canonical model of what we believe enhancers to do. It's obviously very simplistic. The idea being that we have a certain spatial pattern of transcription, in this case, maybe a developing embryo. We have expression in the brain and the forelimbs. The idea being that enhancer A and B collaborate to produce expression of gene X in the right time and place. Now, if the transcription factor in purple can't access that DNA information, perhaps through differential chromatin structure, we lose particular cell type specific spatial, temporal, and abundance patterns of transcription. So of course, p53 though, as I mentioned, is ubiquitously expressed. It's everywhere, all right? Every single cell type we have has p53, and it can be active in every cell type. So we wanted to understand what controlled differential enhancer activity between these two cell types we observed, and between a number of cell types we've subsequently looked at. So an important point about enhancers is that they are actually regulated by numerous DNA binding transcription factors in collaboration with recruited cofactors. So it's this activity of multiple factors that actually leads to what we're looking at, which is enhancer activity and transcription. So we looked pretty exhaustively at these p53 binding sites and epithelial cells to look for motifs that might reflect differences in transcription factor binding. We didn't find anything interesting, at least we didn't think so. So when we looked at motifs found in p53 binding sites that were unique or specific to epithelial cells, we primarily found p53 motifs and motifs for its paralog p63. Now, as a reminder, p63 is restricted in lineage to just epithelial cell types. So we had the question, which is, do these two factors somehow cooperate in order to allow p53 to bind to new locations in a cell type specific fashion? It turns out that's exactly what happened. We have p53 binding to the promoter of RNA 7. We see in red p63 bound across multiple conditions to the exact same region, and this is a region that appears to be a transcriptional enhancer. Now, this is not just at one location, it was at every single unique p53 binding site in epithelial cells had shared binding of p53 and p63. I'm not going to summarize, I'll summarize very quickly all of the results, which is that p53 and p63 collaborated to activate this unique epithelial specific transcriptome. Take a look at the paper if you're interested, or throw some questions in the Q&A, but there's plenty of interesting data here. What we found really interesting is that the reason p53 has all these new binding sites is because p63 is absolutely required for enhancer accessibility and activity in epithelial cells. So if we compare the patterns of histone modifications at p63 binding sites between cells that have p63 and cells lacking p63, I think you can see a strong reduction in enhancer activity, or at least these metrics of enhancer activity at multiple loci. And this matched up really well with a number of publications I'll talk about in a moment, suggesting p63 has an incredibly important role in chromatin accessibility. So to summarize this particular piece of data, or this particular paper, cells lacking p63, there are a number of binding locations that p53 just can't access. The chromatin structure is not in the right orientation such that it can recognize its own transcription factor motif in the DNA. But we have cells that have p63. p63 can convert closed chromatin to open chromatin, and that actually allows a competition between p53 and p63 for a naked DNA motif, and we get this interesting exchange. Now, what I didn't tell you is that we also saw really strong evidence for p63 being inhibitory by p63 just blocking p53 binding sites since they bind to the same sequence. Now, that's something we're interested in looking at moving forward. Now, all of this data came out in the span of about 3 months. We had 5 different papers that all said essentially the same thing, which is that p63 is a pioneer transcription factor and allows for particular chromatin accessibility patterns that seem to be really important for epidermal lineage commitment. So chromatin accessibility alters p53 binding. It alters p53 activity by restricting or allowing access to DNA regulatory information. But p63 is only expressed in one cell type, and p53 is everywhere. So we want to know what else might control p53 activity and control chromatin accessibility beyond just p63. So we used a fairly common approach now, which is the massively parallel reporter assay, where we take a series of potential regulatory elements upstream of a minimal promoter. This minimal promoter drives expression of the green fluorescent protein in this case, but also expression of a unique barcode in the 3' UTR that's linked to this individual enhancer. The idea being that we can screen multiple regulatory elements and read them out by sequencing the 3' UTR associated barcode in that RNA. Now, our idea was we took about 300 wild type p53 regulatory elements. Each had their own unique barcode, and we screened these 5 to 7 times each. We then made a series of mutants that I've binned sort of in this fashion just for simplicity's sake, but we had thousands of DNA variants across this enhancer region. And the idea was to ask if any of these variants, which we would expect would change potentially transcription factor activity, might alter expression of this particular reporter gene in a p53 dependent fashion. Now, I'm not going to go through all of the data. I'll go through one tiny little vignette in my last few moments. But what we came to realize was that p53 activity requires a number of different additional flanking sequence contexts, or a number of different motifs beyond just the core p53 motif. Now, this was apparent at the one we studied the most, which is an enhancer regulating expression of cyclin G1. We can see by evolutionary conservation of vertebrates, the p53 binding site at this location is fairly well conserved, but we also have a very highly conserved SP1 KLF family motif, which looks something like this. When we mutate either the entire region, which we call the R2, or we make individual nucleotide substitutions converting G's to C's, we saw a massive reduction in enhancer activity in any of these cases. And we also saw that happened to be, this enhancer had activity independent of p53. We used CRISPR-Cas9 in vivo to actually make analogous mutations, getting rid of the p53 response element, severely diminished CCNG1 expression, removing the SP1 KLF family motif, did reduced activity, but not to the extent of getting rid of p53, but certainly much more than wild type, or more than an analogous mutation directly next to the upstream or 5' of the p53 response element. Now, the one last interesting experiment that we thought was pretty fun, and I think has some interesting utility beyond just our study, was we used CRISPR-Cas9 to screen hundreds of DNA variants for transcription factor binding within the cell. We made a number of indel and substitution mutations, both upstream within the p53 response element and just downstream in this potential SP1 KLF family motif. And then we used qPCR or amplicon-based ChIP-seq to identify enrichment of p53 binding. So wild type sequences would have enrichment. Any variant that affected p53 binding in vivo would show reduced enrichment in this assay. So ultimately, each dot on this plot represents a single DNA sequence variant that we screened using ChIP-seq. A number of variants in this position in the L2, which is just upstream, showed reduced activity, although none of them seem to be focused on a given motif. Each black dot is a wild type sequence. Each red dot is when we altered the p53 response element somehow. And the blue dots represent times we mutated the p53 response element, but we created a novel p53 binding site. Now, what ends up being very interesting is that all, if not most of, if not all, of the variants that we screened at this R2 position were mutations specifically in this SP1 KLF motif. So we were able to couple CRISPR-Cas9 technology to screen hundreds of DNA binding variants to see whether or not p53 binding was affected by changes in flanking motifs. The conclusion is that this motif, although we don't know the factor yet, unfortunately, there are 14 different factors that can bind to the same motif, all of which are expressed in the cell type. We still are able to say that this motif seems to be really important for binding of p53 to this enhancer. So in conclusion, where we're interested in is that enhancer content and context are critical for p53 activity. We believe a wide variety, and this is all in our recent publications, a wide variety of transcription factors seem to cooperate with p53 to activate transcription. And this happens, we believe, primarily through enhancer elements. There's data in our paper and a lot of really interesting stuff come out from other labs recently. And finally, there is a strong cell type and condition dependence for these factors, suggesting that the core p53 transcriptome is woefully underreported and that there's really interesting cell type dependence that we have not yet gotten to. So again, just thanks to the people involved, and I'm more than happy to take any questions.
Well, thank you very much, Morgan. That was a very edifying talk. I invite the audience to post their questions to the Q&A. For those who've come late, the chat box has been inactivated, so you can't ask questions via chat, but please do so via the Q&A. And there is a question by Hormoz Mazdiyazni, and a very interesting question is, does p53, p73 have similar collaboration as p53, p63?
The correct answer at the moment is we don't know, but I would say it's highly likely that that's true. I'll say that we don't have a lot of good cell models for p73 expression. They don't culture particularly well, although there are some really nice models from Uta Mol at Stony Brook looking at respiratory cell types that see organoids that seem to be really promising to look at this kind of collaboration.
So Hormoz says, thank you, that was a great talk. There's a question from Michael Bradley. Are there any known mutations in p63 that are tied to loss or gain of function of p53 based on their cooperation / competition?
Yeah, I think that's a really wonderful question, because there are obviously a huge number of p53 mutations and a more limited set of known p63 variants, most of which are involved in craniofacial and limb malformations in humans. Now, the question is, are there any known that influence p53 activity? And what I'll say is nothing has been directly demonstrated yet, but in a number of model organisms, we'll say in humans, in a number of model organisms, and flies and mouse especially, a number of interesting developmental defects are actually because P53 is hyperactive. All right, and so we are actually very interested in P60, not only P63's sort of role in promoting P53 activity, but that last little bit in my first sort of vignette, where we noticed that P63 represses P53 activity. So one can imagine developmental defects that come about because P53 activity is too high and cells either die or stop growing or don't hit the right coordinated steps during development. And there's really strong evidence that that is a potential etiology of a number of developmental defects. So I would love to, you know, hopefully that shakes out and people see that, because I think our data would predict that.
Morgan, we have time for one last question.
Okay.
It's from Matt Slungman. Does activated P53 regulate genes more from enhancers than promoters?
Oh, that's a slippery slope question. It's a little bit loaded. So the answer is that we don't know. It's much easier to connect transcription factor activity at promoters to activated gene expression. That's just the proximity really helps make those connections. There's been a lot of really great work though recently, not just on P53, but for example, a number of steroid receptors using things like CRISPR-Cas9 and CRISPR inhibitors to connect distal regulatory element activity to gene function. My prediction, and I think it'll probably hold true from the data I've seen, is that P53 seems to work equally well from enhancers and promoters for the regulation of gene expression. I would say promoters are just easier to identify because they're at the transcriptional start site.
Okay, thank you very, very much, Morgan. We have more questions. We'll save those for later, and you can communicate directly with the people who've asked those questions. And now it gives me very great pleasure to introduce Alan Chen, who is our next speaker. Alan is a newly minted associate professor with tenure in the chemistry department and the RNA Institute. So actually the RNA Institute in Albany is actually a bricks and mortar place in which some of our investigators live, and Alan is one of them. So I first got to know him when he was a postdoc at RPI. I met him at a meeting in Florida and I thought, whoa, this guy is really something. And then decided to try to recruit him to the RNA Institute, and I was successful. So that's a real feather in my cap. Alan is a molecular biophysicist by training. He was trained at WashU. And he uses those biophysical principles to model RNA, RNA folding, RNA protein interactions. And a significant aspect of Alan's work is that his simulations are guided by experiments. And for us experimentalists, we're really, really comforted by that. So, Alan, we're looking forward to your talk. It is entitled Simulating RNA Folding Pathways in 3D Using Sparse Experimental Constraints. And again, I invite the audience to ask questions in the Q&A, and we'll take a look at those after Alan's talk. Thank you so much.
Thank you, Marlene, for that kind introduction. And thank you for all of you out there who've decided to Zoom in with us this afternoon. And I'm happy to share some recent progress in our research. So as Marlene mentioned, my lab is a computer simulation lab. The closest we get through experiments are sort of vicariously through our collaborators. And very much this title slide sort of motivates the grand vision for why we should have computational simulations of RNA. So in a science fiction future, if our models were good enough, it would be absolutely wonderful if one of my biologist colleagues could stop by my office and say, hey, Alan, we just sequenced this really fascinating RNA. Can you tell us what it looks like? And we would sort of sit there and type it in the console and get a cup of coffee and then come back and have some glorious structure spinning on our screen. So that's very much the science fiction, more science fiction than science fact at this moment, but I hope to convince you by the end of this talk that we're making significant strides towards that science fiction future. So why is it that we can't do this? So broadly speaking, the things that hold us back from this can sort of be classified in two general sets of problems. Now, the big problem that me and many other simulation groups have been trying to tackle for many years is worrying that our energy functions aren't accurate enough. Our computers have to calculate forces between all of the atoms in our model, and these forces need to accurately represent the behavior of stacking and base pairing that we expect to see based on experiments. And of course, we've done significant work in this realm as well, but that's not what I want to talk about today, because it is actually a completely separate class of problems than another type of problem, which is on the right-hand side, the sampling problem. So even in the world where we have the most perfect energy function, that a computer could tell us that one conformation is more favorable than another conformation, that's not going to give us the right answer if it doesn't actually sample the right conformations to try. And this is particularly problematic for RNA because there are so many possible conformations. And if you think of a base pair on average adding maybe 3 to 5 kcals per mole of stability, we have this kinetic trapping problem that maybe 100 nucleotide RNA, maybe it has 40 base pairs formed in its most stable state, but there might be many thousands of states that have 35 base pairs formed, and they might be the wrong 35 base pairs. So in other words, to find the ultimate folded state, you'd have to undo some of those base pairs and then try again. And this, of course, happens in real life. This is why structural biologists anneal their RNAs to get them into the most stable state. But of course means that the more realistic our model is, the more of a problem it shows up in our simulations as well, because this could take many milliseconds to resolve itself in real life. And milliseconds is years of computer time on a supercomputer when you have to calculate the position of every single solvent atom, every single salt atom, etc. So this is a particularly deep and rugged landscape for RNA, and we really have to think about how we would tackle this problem before we could look at biologically interesting RNAs. So I promised I wouldn't talk too much about force fields, but I did want to give you a flavor of the direction we've been going. So for the past few years, my lab has been trying to improve the accuracy of these energy functions by looking at experiments that we can calibrate against, preferably thermodynamic experiments that are in solution. So they're as apples to apples as possible. We really want to get the right solubility, the right stacking, sort of anti versus syn, and we want to get all that behavior correct. And we think we've made better models than the ones that were before, as are many other people in the field have been tackling this problem head on. But how do we know that we're actually making progress towards that pie in the sky goal? Well, the proof in the pudding would be the simplest computer experiment that you can imagine, which actually turns out to be not so simple. So if these energy functions were really good, I could just simulate 2 single strands and put them in some water and some salt, and they should form a double strand. And then if I heat it up a little bit, it should go back to single strands, and then we could compare that melting temperature to a real melting experiment, and that would give us confidence that we're getting the right answers for the right reasons. Now, unfortunately, this is almost an impossible problem if you want to have it done at all atom resolution with solvent and ions and everything else. So in real life, it probably takes milliseconds for this to happen. Simulating milliseconds would take years of supercomputer time. Quite frankly, I don't think grant agencies or tenure agencies were going to buy the argument that I should just wait until computers get 10 to the 6 times more faster and we'll have solved this problem. So this provides a big challenge, but what really got us thinking about this problem was a little glimpse into the future. So for those of you who aren't aware, there is a fabulous work by the Shaw's group where they have this special Anton supercomputer which can propagate simulations in time 2 or 3 orders of magnitude longer than the largest NSF supercomputers. And there was some really nice work by Dazi Tan, who was a postdoc at the Shaw. He's now working with Silicon Therapeutics. And they did a similar effort to increase the accuracy of their RNA energy function. And then they actually tried to do what I said was probably almost impossible, which is that they folded a small duplex RNA. So it's incredibly modest. It's 5 base pairs, and that's not a typo, CACAG. So Dazi ran this on his Anton, which is a single user computer and costs, if you have to ask how much it costs, you can't afford it. And he ran this for probably about 6 months. And this is a glass half empty, half full situation because it did fold reversibly. I had always wondered if you had a big enough computer, if that would actually happen. And he proved that it actually did. But the statistics are sobering. It reversibly folded 8 times. And he simulated for 6 months as the only user of a computer that I'll never be able to afford. So this really pointed out that these kinetic traps are so hard to overcome that even with the best computers and even with the computers of tomorrow, it's still going to be a huge challenge if we really want to use simulations as a crystal ball and say we make no assumptions whatsoever, just extend the time axis until we try enough confirmations. So to get around this, we've borrowed an idea really developed by Ken Dill at Stony Brook, where he's been looking at ways to overcome similar problems for protein folding. And the way he thinks about it, normally you're searching this very rugged landscape. You want the computer to decide that this deep minima here is better than all the other ones there through an exhaustive search process. And this could take a very long time. But he points out that if you assume just a little bit of extra information, so say you know that this helix packs against this helix in the folded state, or there's a salt bridge or some other reason for a long range native contact, and you add that as an experimental constraint into your simulation, he calls this narrowing or sharpening the free energy landscape. So basically you've added a fake penalty that discourages the simulation from exploring things that are inconsistent with the experiment. And then it is much less work that the simulation can actually compare the available alternatives and arrive at hopefully the correct solution. Now you have to be a little creative to think about where you would get these long-range native contacts for a protein without having already solved its structure. So maybe you use phylogenies of hydrophobic contacts, maybe you use experimental data from FRET pairs. But for RNA, we have this wealth of information. Even before we type anything into the simulation, we could have run mFold, we could look at shape experiments, NMR experiments. There's so many different things that tell us what is base paired to what, but just not at the resolution needed for a full 3D structure. It may not even involve experiments. It could just be phylogenetic covariation data. If there's a hypothesized long-range base pair from this C to a G 100 base pairs away, and elsewhere on the phylogenetic tree, that G becomes a C and the other C becomes a G, that could already be enough information to tell you that you have a long-range native contact. And the beauty of this is that we don't need enough restraints to completely determine the outcome. The simulation, we don't think they're perfect, but they're still good enough to fold maybe 10 or 12 base pairs at a stretch and come up with that optimal solution. So if you think of what you really want to know for a folded 3D structure, using this nice example from one of my collaborators, Julius Lux, showing the shape chemical probing of an RNase P domain, it wouldn't take a very sophisticated bioinformatics algorithm to tell you that you have 4 long stems here. And I don't want the computer to spend years telling me that P12 is actually a helix. The part that I want the computer to tell me is how this tetraloop interacts with the receptor up here, or how this multi-loop junction comes together. And those can be things that I focus the computation on and leave the boring stuff as sort of restraints that also massively reduces the number of conformations that you want to search. So of course, people have tried many similar takes on this before of adding restraints. And if you just add them without any further ado, you don't exactly solve the problem. So if I add a bunch of base pairing rubber bands to an extended RNA and just sort of fold it, one of the problems is that it's going to crumple into a random ball, and some base pairs will be formed and some won't. But in real life, there's a specific order that they need to form. If they form in the wrong order, you may have to break some of these rubber bands and then try again. And unless you have a mechanism to do that, this type of strategy isn't going to succeed. So our answer to this was to reintroduce an old trick that protein folders have been using for a long time, which is to use replica simulations. So traditionally in protein folding, you would run many copies of your simulation at different temperatures, some above the melting temperature, some below the melting temperature, and periodically they swap and compare information. And that way you sort of get this annealing behavior that allows you to encourage sampling, folding, and unfolding transitions. So what we do is that we sort of have a new twist on this. We add a new axis where we have another set of simulations where we add our base pairing constraints with a dial in front that is varies from 0 to 100%. And this dial determines how strong these rubber bands are for these native contact base pairs. So you can imagine what normally happens in replica exchange is every 1,000 steps or so, all the simulations stop, they all compare configurations, and if someone has found a particularly favorable conformations, it'll swap, and that swap will be accepted 100% of the time. If they find unfavorable conformations, then it's less likely to swap via a Monte Carlo move. So what can happen here is that if your ball randomly crumples up and let's say it finds 2 base pairs but gets stuck, it can't form the 3rd base pair without unfolding and starting again. The replica exchange will provide a chance that it goes to a high temperature, 0 restraint simulation replica where it can completely unfold and try again. And at every point, it's going to be comparing the last best answer to the current answer. So now if it goes through and is able to find 3 base pairs, it will have a better energy than everyone else. It will dominate the simulation until something goes through and can find 4. And if something can find all 4, it's going to be the best answer, and it will come out in an unsupervised fashion. And what's really nice about this is that in the bottom left, we have one simulation that never had any restraints and is below the melting temperature. And we can look at that to understand the natural preferences of the system and to ensure that we're not stuck in conformations that are just there because of how we put the restraints, because the restraints have 0 strength there. So we started by calibrating this on things that had known structures just to make sure that it was an efficient technique. This is a pseudo-triloop from hepatitis B. There's a solved NMR structure from a Swedish NMR group. And I'll mention that this is absolutely painstaking work. The time in between the chemical shift assignment and this final structure here was probably about 4 years. And that's because of the immense amount of effort that was required to acquire enough NOE restraints and RDCs to really nail down the conformation of this structure. Now, one of the caveats of traditional NMR structure solving is that if you get heterogeneity in your NMR ensemble like this, you can't really know for sure that it's from dynamics. This certainly looks like it's a dynamic loop, but the authors are very careful and say that all they can be sure of is that they don't have enough restraints to find one unique conformation that explains all of them. And that is a caveat of these NMR structure solving techniques, which I'll explain in a little bit more in a second. So basically, these were developed maybe 20, 30 years ago, and basically, they are actually simulations. They run a simulation of a fully extended polymer in gas phase with a very crude energy model. They heat it to 5,000 degrees, and then you apply as many restraints as possible. And then the idea is to keep on hammering it until there's only one possible structure that explains all the data. So in other words, we distrust the model so much that we'll trust any experimental data over the simulation, and we'll hammer out any biases of the simulation out. Now, of course, this requires an extraordinary amount of data, which is very difficult in NMR, particularly if you can't resolve all of your peaks and if things are actually moving. So this is very sort of big brother, like, it doesn't matter what the population actually wants. There will only be one confirmation because that is sort of what is predicated. So what we're moving towards is that we can trust the simulation a little more than we did 20, 30 years ago, and we can selectively just use the most informative part of that information, which is that NMR, you can assign who is base paired to what very quickly. So our collaborators at the Karolinska Institute, Katja Petzold, were able to reproduce the basic data required to assign who is base paired to what in probably a few days of work, so much less work than a full NMR structure assignment workout. We can apply those base pairing restraints in our simulation, and then sort of in-house over a weekend, we can come up with a structural model that's very comparable to the published NMR model. Now, I should emphasize that it still is a model. We don't have any restraints on these bulged orange residues here or the loop because there's no base pairing information there. So that's just going to be the preferences of our simulation. It's going to try flipping them in or out and telling us what it prefers, for better or for worse. But we can then just take a time average and say, we actually do expect the loop to be quite mobile. We actually do see flipping in and out, but it prefers to be out in our particular case. And our time to generating a useful model is dramatically reduced from years to possibly a weekend. So you can see here at the end, we just overlay our NMR sort of bundled structure. And this was all recently published in Methods. Now, of course, for this example, we chose something where we knew what the solved structure was, but the real strength of this technique is that we can apply it to things where solving the structure might actually be essentially impossible. So with the remaining time, I just want to tell you a brief story that's about to come out in Nature next week. This is work that we also did with the Karolinska group, Katja Petzold, and they were trying to tackle this very difficult problem of how microRNAs recognize their messenger RNA targets. Now, to oversimplify grossly a lot of biology, it's a single microRNA can recognize many different messenger RNAs by some unknown mechanism via a matchmaker protein, Argonaut, which decides if they're going to get downregulated or not. The bioinformatics easily showed that the first 8 base pairs need to be a complete duplex, but people are not quite sure what happens for the rest of the microRNA mRNA complex, other than that there's a preference for some sort of bulge. People, of course, have valiantly tried to crystallize this, but in the co-crystal structures, the only density that usually shows up are the first 8 base pairs, which are a perfect duplex, and the rest of them are too blurred out to see what's going on. So this intrigued my collaborators' attention. Maybe this is actually dynamic. So doing NMR relaxation studies on a prototypical messenger RNA mRNA complex, she was able to deduce that it's actually flitting back and forth between 2 different conformations about 1,000 times per second with slightly different base pairing patterns. But she was not able to resolve enough distance constraints to actually solve what the structures of those 2 different conformations were. So we took those 2 different base pairing patterns, restrained the simulations of this RNA in our computer, and came up with a surprising conclusion that this 1 base pair difference actually resulted in a 55-degree bend in the helix, which seems gigantic. And of course, that brings up the question, how could such a large conformational change be relevant in the context of the bound Argonaut complex? So what we then did is that we aligned our structures with the seed region, which is crystallographically visible, to match that of the crystal structure, and then try to grow in the rest of our conformations. And to our surprise, it always worked. So the ground state is less controversial. It's well established that it probably drapes across this helix 7 and then goes into this PAZ domain. There's plenty of biochemical evidence that that's the most preferred binding mode. What was unusual was that the 55-degree bend shapes also fit in Argonaut. And the way that they fit is by going into another binding channel that's adjacent to the Peewee domain. And what's really interesting about this is that this is very reminiscent of how prokaryotic Argonauts are observed to bind their double-stranded DNA targets. So it's almost like an ancestral function. And now, while this is a model and we can't definitively prove that this actually happens in the protein, our collaborators were able to do in vitro luciferase assays on a bunch of other targets that they identified that should have the right sequence to do this type of switching as well. And when they mutate them to lock them in sort of the 55-degree bend angle, they actually get an enhancement of downregulation versus the wild type microRNA. So we think this is very suggestive that basically if Argonaut's a matchmaker, one of the things it's looking for is not just if the microRNA likes the messenger RNA, but how well do they dance? Can they access this second binding mode and allow for another way for Argonaut to recognize it? So that's all the time I have today. I wanted to end by thanking those who did the work. Almost everything I showed you today was done by two very talented students, Parisa Ibrahimi, who's about to graduate with her PhD and is looking for potential postdoctoral positions, and Simi Kaur, who actually did this as an undergraduate, who will be starting at University of Wisconsin in the fall for a PhD. Of course, my collaborators, Katja Petzold, and I wasn't able to talk about my work with Julius Lux and SHAPE co-transcriptional folding today. And of course, we're very grateful for our funding, which has allowed my graduate students to expand their culinary horizons from just ramen to now ramen and hamburgers and chocolate. So thank you very much, and I'd be happy to take any questions that you have. Let's see. Let's see, stop share. Okay.
Well, thank you very much, Alan, for a very stimulating talk. There are a bunch of questions. The first is from Michael Bradley. Hi, Alan. Several of your experimental references on the energy function optimization side were oldies but goodies. Are there any currently practiced experimental methods that can help further improve the nucleic acid force fields?
That's a fabulous question. I think that, so what's been really challenging is trying to find a way to separate out the contribution of base pairing and stacking separately. So you can certainly design systems that just stack, like end fraying and things like that. But if you think of your combined Turner parameters that go into your M-fold and things like that, it's always, this is a combined stacking and base pairing parameter. So this is how much base pairing and stacking is together. And it's hard to imagine for me, other than maybe some exotic single molecule experiment, how to separate out just the base pairing from the stacking. That's one of the challenges because it's a very angels on the pinhead kind of argument. In real life, RNA is going to stack and base pair at the same time. But as model builders, we have to come up with some subdivision and say that this much is due to the stacking and this much is due to the base pairing. I think a lot of earlier models went astray because they had way too much stacking, which of course makes your helices super favorable, but then your single-stranded regions crumple up and your tertiary junctions don't work correctly. And I think ways of teasing that out would go a long way. So I think maybe more elaborate single molecule experiments where you can actually pull individual things apart and see what their forces should be would be helpful.
So there's a question from Bruce S. asking if you can model the effects of metal ions and their coordinating capabilities.
That's a fantastic question and one that we worry about a lot. So the monovalent ions, there seems to be good agreement that any reasonable set of sodium or potassium chloride ions we can simulate and they seem to have the right properties. So we tend to try and put in a large excess of monovalent ions, like 1 molar sodium chloride. But of course, we're very cognizant of the fact that real RNAs require magnesium. And this is a huge challenge because in our simulations where we model every single atom, we get what we paid for. And every single atom for magnesium might be 3.5 ions. And then you have to ask the question, well, how do I model half an ion? And then I have this little wrecking ball that goes around and destroys all the structure. And in real life, more or less magnesiums could come from the bath. and we're chained to that. And also the spherical cow model for ions is probably reasonable for monovalence and probably very bad for divalence because they have a preferred coordination geometry. So this is a very active area of research. We're certainly thinking about that as well, that maybe if you add a sphere with some like projections that are preferred ways for it to coordinate, that there's some way that we can include the effects of magnesium without having to resort to some very expensive quantum model. But that's definitely a huge caveat that basically we, us personally, we like it when our collaborators say, oh, it also folds in 1 molar potassium chloride. And we're like, okay, then we have a shot. If it has very specific magnesium effects that we can't use the crystal structure information to place them ahead of time, the chances that it'll find it in the simulation is very small. So that's definitely a very real caveat of simulations.
So we have lots of questions, 8 of them unanswered. We have time for one more from Keith Earl. Very interesting talk, Alan. What are the longest distance constraints that would be useful for your approach?
Basically, anything that it's not really the physical distance that's interesting. It's how far apart in sequence they are. Because if they're very close in sequence and they're just forming base pairs in the helix, then you could have figured that out by other means. But if it's really something that, for example, that shows how 2 helices interact, or how like a receptor or a tetraloop dock together, so sort of the super beyond secondary structure components, then those are the most data-rich things to add in. And those are also the things that your M-fold like bioinformatics programs are least intrinsically able to handle. Right, I mean, just a handful of those, 5 or 6 of those can really nail down a 3-dimensional structure very efficiently.
So, Alan, I want to thank you for a terrific talk. My apologies to other people who've asked questions, but we will save those and hopefully Alan will get back to you. On behalf of the RNA Institute in Albany and its director, Andy Berglund, I'd like to thank you all for participating. I think next seminar of the cooperative is in Sherbrooke or from Sherbrooke. We look forward to that. We wish you all well, and thanks again to the speakers for 2 lovely talks. Bye-bye.
Bye, everyone. Thanks for tuning in.